Grade school photosynthesis is often simplified to plants breathing carbon dioxide and exhaling oxygen. This summary overlooks the staggering chemical problem plants solve every second of the day. In reality, photosynthesis is a high-stakes endothermic redox reaction. It requires a massive, continuous input of energy to forcibly transfer electrons between molecules. The biological goal is to turn airborne gas into solid food. This equation from a bioinorganic chemistry text shows the exact conversion. Atmospheric carbon dioxide is reduced to form energy-storing carbohydrates. Look closely at the left side of the process. To make that conversion happen, the system requires a constant addition of free electrons, specifically billions and billions of them continuously flowing into the reaction. The ultimate bottleneck for plant life is the constant need for a steady, inexhaustible reservoir of free electrons to physically construct the foundation of the global food chain. To cover this massive electron deficit, biology turns to one of the most abundant substances on Earth, ordinary water. Using H2O presents a massive chemical hurdle. Water is an incredibly stable, low-energy molecule. Its atoms are tightly bound together, and it strongly resists giving up any of its electrons. To extract what it needs, a plant has to generate incredible thermodynamic tension. It requires an immense, localized physical force to rip apart the atomic bonds of a water molecule. Splitting water is a feat so difficult that human engineers struggle to replicate it efficiently in a laboratory. Yet plants deploy highly specialized microscopic machinery to tear these molecules apart every single day. To power this microscopic extraction facility, the machinery relies on the energy of incoming solar photons. Plants capture this transient energy using specialized molecular nets called antenna pigments, most famous being chlorophylls. This diagram shows the bioinorganic architecture of chlorophyll. Housed within a wide porphyrin ring sits an essential central ion of magnesium. This geometry is chemically tuned to absorb energy from specific wavelengths of light, particularly the red region. Once photons hit the magnesium center, this trapped solar energy is funneled deeper into the plant's molecular engine. From the magnesium antennas, this harvested energy travels to the primary biological engine, a protein structure known as the reaction center of Photosystem II. Here, we find the actual core bioinorganic catalyst that executes the water splitting. As this chemistry textbook exercise highlights, the predominant metal present at this active site is manganese. The manganese hub drives the oxygen-evolving complex, performing an active chemical violence. It physically strips 4 electrons and protons away from 2 water molecules. This dismantling action breaks the tight bonds, leaving the subatomic components free. This specific manganese complex provides the precise, irreplaceable location where an inorganic liquid is dismantled into the base chemical currency needed to sustain life. Once liberated, those high-energy electrons exit the manganese complex and travel down a chemical pathway, finally fulfilling their role, reducing carbon dioxide into solid carbohydrates. But that leaves behind the remnants of the dismantled water molecules. The reaction strands bare oxygen atoms that have just been forcefully stripped of their electrons. Isolated oxygen atoms are highly reactive. To reach stability, they immediately pair up to form a standard molecule of dioxygen, or O2. For the plant, this resulting oxygen gas is entirely a biological waste product. Having successfully extracted the electrons it needed, the plant simply vents the remaining oxygen out into the atmosphere. Every single breath of oxygen required by animal life on Earth is the direct, ongoing consequence of this microscopic bioinorganic heist. It's a continuous system where magnesium captures sunlight to power a manganese engine, specifically designed to steal electrons from water.